back to my channel. I am İnci, a third year molecular biology and genetics student. Today we are doing another study with me, but I teach the session. I put my iPad away today because I really want to just talk to you. And I really need to talk these objects, concepts out loud to memorize them for my exam. Let's do a quick recap of chapter 1. We learned that our body is under constant attack from pathogens, viruses, bacteria, fungi. To survive, we have a built-in defense system. But today, we are zooming in. We are going to meet the absolute elite special forces of defense system. Today, we are introducing the adaptive immunity and the rules it plays by. To understand the special forces, we first need to understand the regular army or immune system is divided into two main branches. First, we have the innate immunity. Think of this as your frontline soldiers and physical walls, your skin, stomach acid, and generic responder cells like macrophages. The pro, it is fast, so fast, it reacts within minutes. The con, it is completely generic. It hides a piece of wood in your finger the exact same way it fights to a deadly virus. And most importantly, it has no memory. But then we have the adaptive immunity. This is what Janeway focuses heavily on. The con, it is slow. The first time you get infected, it takes about 7 to 14 days for the system to fully wake up, train, and deploy. Pro, it is highly specific and lethal. It creates a custom-made weapon for one exact pathogen. So, what makes this adaptive system so incredible? It relies on 4 fundamental pillars. Let's break them down. The first pillar is specificity. The innate immune system sees a bacteria and says, "Oh, that's a bacteria, let's attack it." But the adaptive immune system says, "That's a Streptococcus pneumonia bacteria, specifically strain number 4. But how does it know? Because adaptive immune cells look for very specific molecular shapes. We call these shapes antigens, which stands for antibody generators. Or adaptive cells have receptors that act like highly complex locks, and they will only bind to one incredibly specific molecular key, the antigen. This extreme specificity means our immune system can target the bad guys with sniper-like precision without causing massive collateral damage to our healthy cells. Second pillar, diversity. But this specificity creates a huge mathematical problem. If we need a specific log for every specific key, how can our body prepare for pathogens that we have never even met? How can I have a receptor for a newly mutated virus that just appeared yesterday? This brings us to our pillar number 2, diversity. Our adaptive immune system has an estimated repertoire of billions, maybe trillions, of different receptors. You have a single cell inside of you right now that can bind to almost any biological molecule that could possibly exist in the universe. How does it do this? Do we have billions of genes? No. We use a genetic magic trick. We have a small set of gene segments and our cells shuffle, cut, and paste them together like a deck of cards to create endless combinations. It's molecular origami. We create weapons for wars that haven't even started yet. The pillar number 3 is the crown jewel of adaptive immunity. It's the reason why you're alive today, and it's the reason why vaccines work. Memory. When the innate system fights a pathogen today, it fights the same pathogen 10 years from now, it learns nothing. Every fight is like the first fight, but the adaptive immune system remembers. When you meet a virus for the first time, you get sick. It takes those 7-14 days for your adaptive cells to find the right specific receptor, multiply, and win the war. But once the fight is over, it leaves memory cells. If that exact same virus enters your body 5 years later, the response is not 14 days, it's rapid, massive, and overwhelmingly powerful. And most of the time, you don't even know you were even infected because your memory cells destroyed the virus before you even felt a fever. Fourth pillar and the final pillar is the most crucial for our survival. Self versus non-self discrimination. Our adaptive immune army is equipped with extremely lethal weapons. They fire at the wrong target, we die. So the system must constantly ask, is this molecule mine or is it an invader? During the early development of these immune cells, the body puts them through a rigorous training camp. If a developing immune cells recognize your own healthy tissues, your self-antigens, it's forced into apoptosis, which is programmed cell death. It is eliminated. When this system fails and your elite army escapes the training camp and attacks your own thyroid, your own joints or pancreas, that is when we develop autoimmune diseases. Distinguishing me from not me is the absolute baseline of immunity. So, who are the exact soldiers performing these incredible feats of diversity, specificity, memory, and discrimination? Meet the two main protagonists of immunobiology, B cells and the T cells. They are both lymphocytes, but they have very different strategies. B cells, think of them as the snipers. When activated, they throw weapons called antibodies that flow through the blood and neutralize threat from a distance. T cells, these are the infantry and the generals. They don't throw weapons. They must physically touch other cells to do their job. Whether this is destroying an infected cell hand to hand or giving orders to the rest of the immune system. We will dive so much deeper into these cells and their molecular shapes in the next part, but for now, let's highlight this chapter and take in what we know about 4 specific pillars of adaptive immunity. And let's move on to part 2. Hello again, everyone. In our last part, we talked about the 4 pillars of adaptive immunity. We said our immune system is incredibly specific, but what exactly it is being specific to? And what is the immune system actually seeing when it looks at a virus? And more importantly, what is the physical weapon that it uses to neutralize that threat? Today, I aim to memorize the molecular structures of antigens and antibodies for my upcoming immunology exams. So, I'm using the Feynman Technique. I put my iPad away and I'm going to teach this to you out loud, just using stories and analogies to make sure my brain actually absorbs it. Grab your coffee and get comfortable. Let's conquer the Janeway chapter 3 together. Let's start with the target. If you read this basic biology textbook, it will tell you that an antigen is any foreign substance that causes an immune response. But immunobiology is strictly molecular. We have to be precise. Strictly speaking, an antigen, which stands for antibody generator, is any molecule that can be specifically bound by an antibody or a T cell receptor. But here's the catch, and this is a classic exam trick. Not all antigens induce an immune response on their own. If an antigen can trigger an immune response all by itself, we call it an immunogen. If it's too small to trigger an alarm, unless it's piggyback another larger carrier protein, we call it a hapten. So remember this, all immunogens are antigens, but not all antigens are immunogens. And now let's zoom in even closer. Imagine a massive bacteria. The immune system doesn't bind to the whole bacteria. It doesn't even bind to a whole spike protein on its surface. The antibody binds to a tiny, highly specific 3D structure on that protein called epitope or antigenic determinant. If the whole bacteria is the criminal, the epitope is the specific tiny tattoo on the criminal's left arm that the immune police use to identify him. So there's two main types of epitopes that we need to know. Linear, or also known as continuous epitopes. Imagine a protein as strings of pairs. A linear epitope is just a sequence of 5 to 8 pairs right next to each other. And even if you boil the protein and stretch the string out, which we call denaturation, the sequence of those pairs doesn't change. The immune cell can still read it. T cells love linear epitopes. Second type, conformational or discontinuous epitopes. Proteins naturally fold up into complex 3D structures. Because of this folding, per number 10 and per number 150 might end up touching each other, creating a unique 3D shape. But if you denature the protein, the ball unfolds and pearl 10 and pearl 150 are pulled far apart and the epitope vanishes. It disappears. B cells and their antibodies are obsessed with these 3D conformational shapes. Okay, this was a long introduction and my brain is already warming up. We have already successfully identified the target, the epitope. But how do we catch it? How do we handcuff this criminal? We use the most elegant, flexible and deadly protein in the human body, the antibody or immunoglobulin. For the next part of this part 2, I need to visualize and explain the exact structural architecture of an antibody. This is the heart of immunology. If we don't understand the physical shape of this weapon, we can't understand how vaccines or monoclonal antibodies or allergy treatments work. Let's build it together. And let's build an antibody together. Antibodies are glycoproteins secreted by activated B cells. The basic structural unit of an antibody is a Y shape, literally like the letter Y. It is made up of 4 polypeptide chains. Let me find a fan. Okay, 4 pens. Maybe 6. I found them. Okay. I found them and let's go with this. Do you see them? This antibody, please imagine it like a Y shape because I can't hold 6 pens in my hands. 2 identical heavy chains. These are the long... It's gone. So, let's recap. 2 identical heavy chains. These are the long inner parts of the Y. They go all the way from the tips to down to the bottom of the stem. 2 identical light chains. These are shorter outer parts that only sit on the arms of the Y. How do they stay together? Through molecular superglue. disulfide bonds. Disulfide bonds. These sulfur to sulfur bridges hold the heavy chains together and pin the light chains to the heavy chains. This makes the structure incredibly stable in the harsh, turbulent environment of your bloodstream. Let's dive into the top of or why. And let me drop these. We divided the Y horizontally, and at the very tips of the arms where my fingers are touching, these are called variable V regions. But why is it variable? Because this is the exact part that grabs the epitope. Since there are billions of different epitopes in the world, we need billions of different tips. This is the log to the antigen's key. The stem and the lower arms called the constant C region. The constant region doesn't care about the antigen. Its job is to talk the rest of the immune system. Once the variable region catches a virus, the constant region acts like a flag, waving at the macrophages, saying, "Hey, I caught something, come, eat this, eat it." And if you're studying like me from Janeway's book, you might get an exam question about papain digestion. Scientists actually figured out this Y shape by cutting antibodies with an enzyme called papain, acting like molecular scissors right at the hinge. When you cut the Y in the middle, it falls into three pieces, the Fab fragments, fragment antigen binding regions. These are two separated arms. They still bind to antigen perfectly, but they can't call for backup. Not these, I guess. I don't remember which one. Fragment antigen binding regions. But they can't call for backup because they lost their stem. And one FC fragment crystallizable region. This is the stem. It can't bind to antigen at all, but this is a crucial part that interacts with the immune cells. Macrophages have FC receptors on their surface specifically designed to grab onto this stem and connecting the arms to the stem. This is the hinge region. This gives the antibody flexibility. Pathogens aren't perfectly flat. This hinge allows two arms of the antibody to swing open and reach two different epitopes at the same time, like a molecular gymnast. Wow, okay. That was a long part too. Let's do a quick wrap up. We started with the target antigens. We zoomed in to find the specific binding site, the epitope. We learned that the T cells like the straight, linear pearl strings, while B cells look for 3D folded shapes. Then we built the weapon, the antibody. We constructed the four chains, heavy and light, pinned them together with disulfide bonds, and divided them into variable region, the log, and the constant region, the FC fragment stem that calls for backup. So I feel so much better about this part right now. Explaining it outline makes me understand it better and forces me to visualize every single molecular bond in my head. And I hope this deep dive helped you visualize it too. So if you're ready, let's take a deep breath and move into the final part, the part 3 of this video. And let's take a break. Then I'll see you in the part 3. Hello everyone, this is part 3. In last part, we talked about those beautiful Y-shaped weapons floating in our blood. But here is a terrifying truth about antibodies. They are completely blind to what happens inside of a cell. If a virus manages to sneak inside your lung cell and closes the door, the antibody is useless. It can't get in. To survive, our immune system created a different kind of soldier. A soldier that just doesn't look at the outside, but demands to see what is happening inside. And it is called the T cell. And I will try to teach you about these T cells and why they are the ultimate divas of the immune system. What is the major histocompatibility complex (MHC), and we will end with the golden rule of the immunology, rule of 8. Let's dive in chapter 5 and chapter 6 of Janeway's Immunobiology. Let's compare our two protagonists. The B cell is the free spirit of the immune system. A B cell can look at the whole intact floating COVID-19 virus, grab it, and say, got it! It sees a raw, native 3D antigen. But the T cell receptor, TCR, is incredibly high maintenance. It strictly binds to the free floating antigens. If you put 1,000,000 T cells in a test tube with 1,000,000 intact viruses, absolutely nothing will happen. They will just float past each other. Why? Because T cells are MHC restricted. Think of it like a restaurant. B cells are like wolves in the wild. They will hunt and eat raw whole chicken. That's the intact antigen. And the T cells are like Michelin-star food critics. They refuse to look at the raw chicken. The antigen must be taken into the kitchen, chopped up into tiny linear pieces, cooked and presented to them on a very specific personalized silver platter. And that chopping process in the kitchen, that is called antigen processing. And that silver platter, that is the MHC molecule. And the waiter bringing it out, this is the antigen presentation. If there is no platter, the T cell critic goes home. It is completely blind. Okay, so T cells need the silver platter. Now, we have to talk about the platter itself. This is where this topic gets really heavy. The MHC, major histocompatibility complex. In humans, we call this the HLA system. If you ever watched a medical drama and they talk about finding a match for an organ transplant, they are talking about matching those exact MHC molecules. Your MHC genes are the most diverse genes in your entire genome. My silver platters look completely different from your silver platters. They are your biological fingerprints. But evolution didn't create MHC just to make an organ transplant difficult. Evolution created them because viruses hide. We needed a mechanism to force our cells to put a billboard or a window on their surface, showing the immune system exactly what is happening inside. And depending on the emergency, we have two different types of billboards, MHC class 1 and MHC class 2. This is the climax of adaptive immunity. If you understand this, you understand immunology. Imagine MHC1 as the internal security camera of a building. Where is it? It is on almost every nucleated cell in your body. Your liver cells, skin cells, heart cells, they all have MHC1. What does it do? It presents endogenous antigens. It constantly takes samples of proteins being made inside in the cell, chops them up in a molecular garbage disposal called proteasome and displays them on the surface. The scenario: if a cell is healthy, it displays normal self-proteins. The T cells walk by and say, "Cool, nothing to see here." But if a virus infects your lung cell, the cell will chop up viral proteins and display them on MHC1. It is basically raising a red flag saying, "I am infected from the inside, kill me." Now MHC2 is completely different. This is not for every cell. This is the exclusive VIP intelligence report. Where is it? It is only found in professional antigen presenting cells, APCs, dendritic cells, macrophages, and the B cells. But what does it do? It presents exogenous antigens. These professional cells patrol your body and find the bacteria floating outside, eat it, digest in their acid stomachs, and present the pieces on MHC class 2. Scenario: the macrophage isn't infected. It is acting like a detective. It holds up the MHC2 platter and says to T cells, "Hey commanders, I found the trash in the tissue. What are your orders?" And we are at the 40-minute mark. I don't know what timestamp we will land on the video after I cut out the bloopers and the waiting parts, but stay with me, hang on there, because we will revisit our first basics of immunology lesson again. So we have two types of MHC platters, and guess what? We have two main types of T cells to read them. CD8 cytotoxic T cells. These are the assassins. Their job is to kill infected cells. And CD4 helper T cells. These are the generals. Their job is to secrete chemical messages to organize the whole army. But how do they match up? How does the assassin know not to kill the detective? We use the rule of 8. The product of the CD number and the MHC class must always equal to 8. CD8 T cells bind to MHC class 1. 8 times 1 is 8. Why? Because MHC1 says, "I am infected inside." So CD8 assassin binds to it, releases toxins, and forces the cell to commit apoptosis. The problem is solved. And CD4 T cells binds to MHC class 2. 4 times 2 is 8. Why? Because MHC2 is on a macrophage holding an intelligence report. The CD4 general reads the report and says, "Activate the B cells, send more macrophages." It orchestrates the war. If a CD4 general tries to bind an MHC1 infected cell, 4 times 1 is 4, so it doesn't work. The biological handshake fails. Math must be equal to 8. This beautiful, simple math ensures the right soldier responds to the right emergency. Wow, we just covered huge 2 chapters about immune system. If you're still watching this, you're amazing. We started this part with the picky T cell receptor that refuses to look at the raw antigens. And then we moved on the silver platters MHC class 1 for internal security and MHC class 2 for outsider intelligence. And in the final, we connected it all with a beautiful, elegant rule of 8. And I finally feel like I understand this story a bit more. Instead of just memorizing it out loud for my exam, I told this story and I shared it with you, and I really get to understand better. So I really hope this deep dive helped you as well. Please let me know in the comments if you survived this whole 3 parts and did this analogies make sense? Drink lots of water. Give your brain a rest. Everything I mentioned in this video will be on the description box down below. Don't forget to subscribe and hit the thumbs up button as well. And I'll see you in the next study session. Bye guys.